Welcome, everybody, and of course, anybody else who wants to join, please do. Some of the basic rules. So first of all, I'm delighted to see you all and happy. I know it's holiday season for many of you, so I'm grateful for you guys taking the time to meet with us. Some of you have sent questions, so I've put together a list, and I'll try to address the questions that you've sent us. And then you're all live, and so if you want, you have the option of raising your hand. And I have two colleagues here, Michael, who you know, and Anat, who's been working in developing this course from the very beginning. And they to them in chat. They will be able to see if there's anything that you would like to ask right now, alive. And we'll see how we can proceed. So I suggest we begin. I have a list here. And the first question I would like to address, I've tried to consolidate some of the questions that might have some common themes about them. And the first, excuse me for not knowing necessarily your first names, but according to the link address, Mark the Virus asks, how long do viruses survive outside of the body? How resilient are viruses? So first of all, let me make the distinction, I'm sure you know that there's a very significant difference between viruses and bacteria. In bacteria, they have the capability of replicating outside of the body. They have a complex sugar, lipid, protein coat or cell wall. And so viruses tend to be very resilient. They have that opportunity of replicating. Whereas, as we've mentioned, viruses being the ultimate parasites, they have no ability to replicate at all outside the cell they have infected. Bacteria can sometimes survive days, months, and sometimes even years. Bacteria also have the ability to create spores. And these are structures that give them very long survival rates. And we know, for example, that anthrax can last years in a dormant state of a spore. And then it can be revived and ultimately be infectious, even years after the original cells were multiplying in sheep or wherever they had been. Viruses, on the other hand, and that was the essence of your question, don't have a cell wall. They don't have the ability to replicate outside of the cell they've infected. And so, of course, their survival is rather limited. And so, generally speaking, I would say that viruses outside of the cell can last anywhere between minutes and possibly hours, but not very much longer than that. Now, the duration is very much dependent on the surroundings and the certain conditions. First condition, of course, would be humidity, because as you realize, if you happen to sneeze into some tissue tends to be dry, it's absorbent. So porous and absorbent surfaces tend to reduce the survival time of a virus markedly. If, on the other hand, droplets tend to fall on something which is smooth, glass or plastic, then the rate of dehydration, the removal of water from the virus, would be less, and so their survival would be longer. Obviously, if it's humid outside, if it's cooler outside, then the survival might be longer, and you might see viruses that can exist and be infectious even hours, maybe a day, but really not much more than that. There are other factors, such as, of course, if it's hot outside, proteins tend to denature. So if it's very hot, we may see denaturation of some of the viral proteins. If there's strong UV irradiation, sunlight, then that may be damaging to the viral genes, whether they're RNA or DNA. RNA and DNA are sensitive to UV irradiation. And then finally, as far as other factors that can affect the survival rate of viruses, could be the nature of the surface itself, not only being absorbent or not, but for example, stainless steel is actually a surface where viruses can survive much, much better than were the surface, for example, copper. And so I think that more or less gives you a feeling. Bacteria can survive for days, months, and possibly years under certain conditions. Viruses anywhere between minutes and hours routinely. And again, it's very much affected if it's absorbent surfaces, if it's hot outside, there's UV, and so on. So I think that more or less deals with the first question that we've dealt with. Now, Nuru Muhammad DP. Again, I apologize for possibly not knowing exactly the name, but I had three questions. And one was, what affects the ability for a virus to be necessarily airborne? And the question was, is that airborne virus more prone to be airborne if it's small, so size, or if it's lightweight? And frankly, I think that we have to forget about what we understand and perceive in the physical world. Viruses are amazingly small. So I don't think that in comparison to the size of droplets, which are huge compared to the minute viruses, the size of the virus per se has very little to be about whether the virus is going to be airborne or not. Much more important is the accessibility of the virus to our oral, nasal, and lung cavity. Obviously, if the virus replicates in the lungs, such as in the case of flu or measles, then there's an abundance of viruses in the fluids of our lungs. It's there, it's physically present because that's where the viruses are replicating. And then under those circumstances, if a person then sneezes or coughs, he generates droplets which contain the viruses that were in the lungs from the very beginning. It's something like what we discussed as far as mosquitoes, that when we ask, is a mosquito bite necessarily infectious? And the reality is this, that if, for example, HIV does not replicate in the salivary glands or in the gut of the mosquito, then a mosquito bite will not be infectious for HIV, as opposed to yellow fever, as we described. So if I summarize that aspect, what makes a virus, let's say, susceptible for airborne, then the first condition would be is that that virus has to be able to replicate in the lungs of the infected person or animal. Now let me see. The same student asked the question as to what constitutes an infectious dose. Is one virus enough, or do we need a lot of viruses to be infectious? So then this too is a question which can be rather complicated. We have 2 points that we have to consider. If there is a preparation of viruses, some of the viruses may have dried out, they may have been damaged, and may not necessarily be infectious. So that being the case, we have the tendency of using a term which compares the actual number of particles, of virus particles in solution, and how many of that, what fraction of the particles are actually able to infect a cell or a plaque-forming unit or cause CPE, cytopathic effect. And so that is one ratio which is important. How active, how functional. Yes. Oh, hey, Ron.
Hello.
Wonderful. Hi, how are you doing? Let's see where you are. One second. Here you are. Here.
And I'm
here.
smiling.
Smile over there. Here. Iran is smiling. You all know Iran. He's our favorite helper here, Professor Iran Erwa Bachach. And thanks for dropping in.
So have fun.
Okay, thanks a lot.
Bye.
Bye. See you, Iran.
See you.
So we have a surprise visitor, which is always nice. Iran is a professor of virology here at the university and has helped me very much in the course of putting together this course. So we were at the question of infectious dose. And infectious dose, first of all, we have to know how many functional particles that are able to infect cells exist in a given preparation of viruses, and so that's less. But the question I think that you're most concerned about is how many viruses do we need to actually cause or elicit disease? And this can change because some viruses are very virulent, very infectious. As Professor Baltimore mentioned, that measles is a very infectious virus. We know that typically in a solution, it might be thousands and tens of thousands of viruses that we need in order to actually cause a disease. And this can change for various viruses. Some viruses are more infectious than the others. Finally, let me just return to my list of questions. I think we've dealt with these questions that were asked about the infectious dose. And now I would like to address some questions that arose from flu. There were a number of you that talked and asked about flu, Monica on the one hand and Jenny on the other. Is that you, Jenny? You can wave if it is. Jenny, hey, hi, how are you? Good to see you.
Can we unmute her?
Do you want to say something, Jenny? You can be unmuted if you would like, or I'll just try to address the question that has been asked. Actually, I see that Monica and Jenny asked about HIV. Is that true? Jenny, are you the one that asked about HIV?
No, I asked about the reverse transcription.
Okay, so that's what I'm referring to. The question was, and it's actually a very interesting question, is that we know that HIV is a plus-strand RNA virus. And so from other examples of positive strand RNA viruses, the notion is that they can be used immediately as messenger RNA. And so then the question that Jenny asked was, if that's the case, why does HIV necessarily have to use reverse transcription? What is the advantage, if there is any advantage, in taking positive strand RNA and making a copy of a double-stranded DNA rather than going directly to replicating with the positive strand RNA? And there are a number of points that I would like to make. First of all, in biology and molecular biology, we don't always know the answer as to why things develop, but we are able to say that this is a fact, that they developed as such. And so then as biologists, we ask, does this afford any advantage to the virus because it exists as such? In the case of HIV, the fact is that it's a retrovirus. The positive strand RNA is reverse transcribed into a double-stranded DNA provirus, which goes into the nucleus and is integrated into the host cell DNA. Now, think about it. When that occurs, when that infected cell divides, the daughter cells already have HIV genes in them. And so in reality, that affords a second route of infection. Each time the infected cells divide, these cells already have in their genome, in their DNA, HIV genes. And because they already have genes, it goes on from generation to generation without the virus ever having to leave the cell. So this is one advantage. It affords a new route of infection, not virus infecting the cell, but from cell to cell division, the virus can propagate. Now there's another aspect. It turns out that the process of going from RNA to DNA and then back to RNA with the cell's polymerase is actually very error prone. Now, as human beings, we would say that maybe it isn't such an efficient virus because it's making a lot of mistakes. But when you think about it, a virus that tends to generate many, many mutations and variations, these viruses have a survival efficiency because they can evade immune surveillance. They can constantly change. So if, for example, a given virus is no longer recognized by the immune system because of these mutations, it has a chance to survive. And so evasion of the immune system is increased if the virus is constantly changing. So these are some of the reasons, or at least advantages, that might be born through the fact that HIV is a retrovirus and it has its unique mechanism for replication. And I hope that gives you some sort of an explanation, Jenny. There was another aspect of HIV that I wanted to mention, and that is in lesson 6. So please stay on with me to lesson 6. Lesson 6, we'll be discussing HIV specifically. And in fact, one of the co-discoverers of HIV, Professor Bob Gallo, will be our guest in lesson 6 and discuss the importance of blood tests for HIV and others. But it's interesting that HIV is also the product of zoonosis. There is a virus in monkeys called SIV, simian immunodeficiency virus. And this virus exists in monkeys, in African green monkeys in Africa. And when it infects the monkeys in Africa, these monkeys don't get sick because through the years of evolution, this virus has come to some sort of a equilibrium and the ability to live with the monkey without causing disease. However, it's curious. If you take the African virus, the one from the monkey, and infect an Asian monkey like rhesus macaque, these monkeys now all of a sudden suffer from severe immunodeficiency. Now, why is that interesting? Because it says to us that in different hosts, viruses can be causing disease or not. We know the bats that may be carrying Ebola don't necessarily suffer from hemorrhagic fever or from a serious disease. And as scientists, we're very curious about why is a virus so disease-causing in one organism, and on the other hand, actually living in peace, in synergy, or without causing disease in another organism. And we study these things. Now let's go to flu. In flu, let me just see who are the students that asked. Okay. Some bashful student asked about flu. There are 2 questions which I will relate to in flu because it's interesting, but they were submitted by somebody who calls himself Anonymous. So, okay, so Mr. Anonymous, you can write to me, maybe tell me who you are, but that's fine, but the questions are valid nonetheless. One question about flu is if I would be able to explain how the transition from the flu virus moving from birds through pigs and to humans, how does that work exactly? And so the reality is this. We mentioned that flu viruses replicate in birds, and they have their own receptor. And the receptor is a sugar, it's sialic acid, and the link in that sugar receptor is alpha-2, 3, that is connecting carbon 2 to carbon 3. And this is typical of the bird receptor. When the flu virus emerges from birds, it knows how to bind the bird receptor. And if it happens to be taken in by a friendly pig, in the lungs of the pig, there are cells that have exactly the same receptor, the alpha-2, 3. But pigs are curious in the sense that they have cells that have other receptors as well. And they have a structure of sialic acid, which is 6-2 or 2-6. And so because both receptors are in the same lung, when flu viruses replicate in the lungs of pigs, they encounter both receptors. They see some cells with the bird-like receptor and some cells with the human-like receptor, the 6-2. Now, if the flu virus happens to undergo mutations, then there may be some flu viruses that have emerged from a cell of bird receptor, but it's been modified a bit. And if it's been modified to the extent that it now has the ability, fortuitously, to bind the human-like receptor, it may infect that cell. And of course, viruses that emerge from that cell are all going to have the ability to bind the human receptor. And so in the flu, in the pig's lungs, you have a mixture of cells that represent the human-like receptor and cells that represent the bird-like receptor. And so a mixture of viruses occur as well. Those that are able to infect pigs via the bird receptor, as opposed to those that can infect via the human receptor. Viruses that emerge from cells that have human-like receptors, if they form airborne droplets and are taken in by a neighboring human, chances are those viruses will now be able to infect humans, and so on and so forth. So the reason why pigs are a good intermediate is because they have both receptors simultaneously, and mutations within the virus can actually generate the ability to cope with one receptor as opposed to the other. Now, the second question by Mr. Anonymous or Mrs. Anonymous dealt with what and how different are the various groups of flu. We know that flu virus, and we'll talk about this in lesson 7 in flu vaccines, we may learn about the flu vaccines that every year the World Health Organization, the WHO, determines what the composition of flu vaccines is going to be for the coming fall. And there's constant change in the flu virus composition of vaccines. But the question that was asked is, if there's so much change, how is it that there's only, let's say, 16 or 17 H1 type of receptors? If there's a lot of variation, there should be H1, 2, 3, and maybe H1000. And the reality is this: within the family of H1s, there's constant genetic variation, but not sufficient to make it a unique and distinct hemagglutinin. For example, the H1 hemagglutinin is 566 amino acids long. And it still has the general traits and characteristics of H1, even if there are 10 or 20 different changes in the amino acid composition. And so, provided that the H1 protein in general looks like H1, there are 566 amino acids, and from year to year, there are variations of somewhere around, I would say, 20, 30 amino acid changes, one still considers them H1. So they're constant changes, but they're not enough to actually cause a difference. So then you'll ask, so what about H3? How different is that from H1? And in H3, I would say that there could be as many as 350 amino acid changes as compared to H1. And so if that's the case, that is now designated as something significantly different. It's no longer H1 because of the very significant number of changes in the amino acid composition. I see that we have a bunch of questions in the chat, and so I'll now take a look to see, and this is from Mavi. Let me see where you are. Do you want to unlink or unmute Mav so I can hear? And what about I see? And You have a camera?
I have.
Why don't you turn it on so I can see who I'm speaking with? Come on, don't be bashful. Jenny, where are you from?
Australia.
From Australia, from down under.
Yeah. Sydney.
Okay, wonderful.
It's 2:30 in the morning.
Oh, I'm sorry about
So
that.
I'm very keen.
We tried to find an hour that would be somewhat in the middle.
Yeah,
But
I
great.
understand. Yeah.
Okay, great. I'm glad to hear that. So, Maui, where are you from?
I'm from South Africa.
Oh, great. So,
So we're on
I
the
see
same.
we have a lot from the British Empire of past. Israel was also a mandate, so that's fine. So, do you have a camera, or should we just do this straight on? Oh, here you are. Great. Wonderful. So why don't you ask your question directly? What did you want to ask?
I was a bit interested in the varicella, the chickenpox.
Varicella?
And why does it go into, or why can it cause herpes zoster later on in life? Is it the zoster part that plays a role here, or is it is varicella that's related to herpes, or are the herpes viruses all in the same group?
Okay, so I'll try to address that. First of all, you guys have to realize I'm just a scientist.
Okay.
I'm not a doctor, okay? That is, I have a doctorate, but I'm not a physician.
Yes.
But I'll do my best.
Okay.
Varicella zoster is a virus. The virus itself is of the herpes family, so it's a DNA virus. It has a very, very large genome, maybe 200. Hey, I see the young child in the picture as well. That's good. Hi. Oh, 4. My God, wonderful. I've got 10 grandkids, so that's okay. Great. He's 4. So in any case, chickenpox is caused by varicella zoster, this herpes. And when we get better, the fact of the matter is that we don't actually clear the virus completely from our body. So although we recover and we no longer have lesions and scabs and so on, and we're no longer contagious, some of the virus goes underground in our nervous system and sits there for many, many, many years. Now, of course, the experience of being infected by the chickenpox virus, by varicella, induces a very effective protective immunity. And so we'll be learning about how we store our memory in lessons 5 and 6. So next week, you'll be starting with the innate immunity. And then in lesson 6, you'll learn about how we store memory cells, B and T cells. So the bottom line is that although you've recovered, you have reservoirs of infectious chickenpox virus in our bodies. We're just saying they're latent. They're sort of laying low and not causing any disease. However, as we get older, we undergo a process which we refer to as immunosenescence. Our immune system tends to be less and less infected until you become old like me. And so if that's the case, people who are above the age of, let's say, 60, 65, and I qualify for both, our immune system is not quite as effective as it had been when we were younger. And so in view of that, the virus, which I don't think we actually know why it emerges again, but when it does, we don't have quite the same strength of memory and ability to subdue it. And so it begins to infect along axons, and that is what gives us shingles in later life. So in reality, the whole phenomenon of shingles in later life is relevant to those people in childhood who have been infected by the virulent and infectious chickenpox virus. Recently, a number of companies have come out with an alternative vaccine for older people, and I've been vaccinated. And that alternative vaccine is a higher dose of the live attenuated virus. We'll be talking about that in lesson 7, how vaccines are made. But the vaccine that we get for shingles is in essence exactly the same virus of chickenpox, but at a higher dose, and it's given to the older people's population. So the question is, for example, if a child has been vaccinated, will he develop shingles from the vaccine in later life? And the answer is definitively no, because the vaccine doesn't cause chickenpox. The vaccine is a virus, it's an attenuated virus, but it does not cause disease. And so even if it were to emerge in later life, it would continue not to cause the disease. So that's good. Then another question I've been asked about varicella zoster and shingles is, if somebody has been ill with shingles, should he get vaccinated nonetheless? And the answer is, after he's fully recovered and maybe a half a year to a year later, yes, that would be a good idea. So I hope that answers your question about shingles, varicella zoster, and everything you wanted to know about the older population.
We have another one.
We have another question here.
No, we have another child from Vaya. Her child is in.
Yeah, I noticed that we have, and what's her name?
His name is Daniel.
Barniel. I'm envious of him because I see he's got much more hair than I do. Excellent. Good. Good. So are we going to continue with some of the questions? We talked about retroviruses, and we talked about how long viruses are infectious, and we talked a little about HIV and flu. And then there was a question by 3141PI. I don't know if you're here today, and if not, it doesn't matter because that sounds like R2D2. In any case, the question is are we expecting any new emerging epidemics in the near future? And are there possibly any viruses that emerge spontaneously in people? Now, I asked a number of people, amongst others, our friend Eran, and I really don't know of any example of a virus that emerged in humans totally spontaneously. Most of the viruses that I'm familiar with have in some fashion or not developed over years through zoonosis, although there are quite a few viruses that are uniquely human, for example, polio, but I don't know where it came from. But we do see a constant emergence of diseases and epidemics that are derived from the animal kingdom that infect humans. Now, of course, probably the most famous event that occurred over the last few decades is HIV. And I mentioned that previously when I was talking about HIV/AIDS. You know that there are similar viruses in the monkey population, and the origin of HIV probably stemmed from either chimpanzees or African green monkeys. And so we expect that this happened a number of times during the history and evolution. But most recently, and actually it's very relevant to our geographical area of Israel and the Middle East, because as you may recall, maybe 10 years ago or 15 years ago, there was a very serious threat about a lung disease called SARS. And this was a disease which emerged out of the blue. Nobody knew about SARS. And it is a type of virus called a coronavirus. And this disease causes severe lung disease. And people were very concerned about the epidemic. Something like Zika that Scott Weaver, Professor Weaver, discussed as how sometimes emerging epidemic viruses travel on airplanes from one passenger to the next. And with SARS coronavirus, we began to see a disease stemming from China, but ultimately coming into Canada through air flights and so on and so forth. But as very often happens, these viruses tend to peter out and become less of a threat over the years. Now, why am I mentioning SARS? Because it's this coronavirus, and actually in our lab, we've been working on SARS for quite a few years. But as it petered out, less and less people were interested in this particular virus and how dangerous it may or may not be. And so we more or less put that research on the back burner. However, just a year or two ago, a cousin of SARS, also a coronavirus and also a very severe lung disease, emerged, and it's called Middle East Respiratory Syndrome virus, or MERS. And this virus, the index patient, you remember we talked about the fact that the index patient is the first known clinical case of a given disease and virus. And so the index patient was in Saudi Arabia. And this virus began to spread throughout the Middle East. We're talking about Egypt and Jordan. Fortunately, we haven't had any cases in Israel, but there have been quite a few cases throughout our area of the world. And then all of a sudden, the same virus appeared in Korea. And so we're seeing the same dynamics of an epidemic. It's a serious disease. It infects the lungs. It causes a respiratory syndrome. And in addition to that, it's spread throughout the Middle East on the one hand, and now it's in Asia. And there are quite a few people who are concerned about, will this virus develop into a very serious, life-threatening type of epidemic. Now, the interesting and curious part of this particular disease is that it is zoonotic as well. It's believed that maybe the original reservoir of this virus was in bats. But what we know for sure is the reservoir here in the Middle East is none other than camels. And so here's a curious situation whereby we have camels that were possibly infected by bats. And then subsequently, these camels have infected people. And now we're seeing the emergence of an epidemic called MERS coronavirus. And there's something else that you wanted to tell me? I'm consulting with the team. I'm not alone here in my office. Yes.
If people want to say hi, we can open the cameras and microphones.
Okay, are we just about finished? I see. Okay, so what we're going to do is this. If there's anybody who wants to say hello, introduce themselves, or ask a question, fine. We have to stop everything in a minute, just 1 second. And then if you continue to write questions to me, I'll see if I can address them subsequently in the forum, in the website.
Hello.
You're unmuted by the host. Mark?
Oh, hello. Well,
Hello.
just to thank you for your time, and it was very enlightening and very interesting response. Thank you very much.
Very good. Mark, where are you from?
Switzerland.
From Switzerland. Good. And where in Switzerland?
Near Lausanne.
Near Lausanne. Okay. Obviously,
So
I can
the
hear
French
from the
speaking,
accent.
yes, my accent.
Grüezi miteinander.
Danke sehr.
Yes. Very good. So I think with that, I want to thank everybody very, very much. It's been enlightening for me as well. And good luck to all of you as you continue with the course. Continue to write and ask questions, and we'll do our best to respond. So take care. Have good holidays. Happy New Year. And continue to enjoy viruses in theory rather than in practice. Bye.